In this episode, we are joined by Dr. Erica Korb from the University of Pennsylvania, whose research investigates the intersection of epigenetics, chromatin biology, and neuroscience. Dr. Korb explores how dynamic regulation of histone modifications and chromatin architecture influences gene expression in neurons, shaping processes like learning, memory, and synaptic plasticity. How does the epigenome adapt to chromatin remodel as playing cognitive function and neurodevelopmental disorders? Join us as we uncover the epigenetic mechanisms that encode experience into the human brain.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
So I'm happy to talk to you now, Erica. Welcome on the show.
I'm happy to be here.
So without a lot of small talk, I guess we can start with the question. So a question I like to start off with and to ease into the topic and into the podcast is, how did you become interested in biology in the first place and then pursuing a career in science?
Great question, and a fun one to start with for sure. I was in college and I love science. And I was trying to figure out what to do with that general interest. I didn't really want to be a doctor because I wanted to actually kind of do the science. I didn't want to sort of just apply it. And I was interested in both biology, specifically neuroscience, and I was also really interested in astronomy. But it turns out I was really bad at physics, and you need a lot of physics for that. But I was pretty good at chemistry and biology, so that kind of won out in the end. I also was really lucky to get to do a summer internship in a lab kind of early on. And the first time I looked through a microscope and saw neurons fluorescing with some beautiful antibody stains, I was completely hooked. It was one of those moments where I was just in awe. I was sort of aware of the fact that I was getting to see something and sort of solve a problem that no one had figured out before in the history of humanity. It was a very tiny problem, and it was not a very, hard experiment or anything like that, but just that moment of discovery paired with sort of the beauty of these neurons under a microscope was enough to really catch me and sort of lead me down this path.
I also can remember that the fluorescent staining of cells is like really something that is very fascinating when you see it the first time.
It is. Seeing is believing, I think, for a lot of science, and it's one of those real visual pieces of data. Obviously, it has to be paired with many other kinds of analyses, but it still always has a place in my heart as far as experiments go.
Definitely. So, for those unfamiliar, could you introduce yourself and your current role?
Certainly. I'm Erica Korb. I'm an assistant professor at the University of Pennsylvania in the Perelman School of Medicine. I have a primary appointment in the genetics department. I'm also part of the Epigenetics Institute here. So our main focus in the lab is working on epigenetics in the context of neuroscience. We're particularly interested in histone biology and mechanisms that control gene regulation in neurons. We have a focus on neurodevelopmental disorders, such as disorders that lead to autism spectrum disorder or intellectual disability and other related phenotypes. But we also spend a lot of time just asking really important basic science questions as well.
So you just mentioned it, that at UPenn you have an epigenetics department. So epigenetics is very strong at UPenn, right?
It is technically Epigenetics Institute, but it's
Sorry for that.
very a very large group of faculty all coming together with this common interest, and it's a large number of different labs all working on kind of every aspect of epigenetics you can imagine. We have regular meetings, symposiums, retreats, seminar speakers. It's a really vibrant community. And it's actually one of the reasons I chose to come to Penn is because of that epigenetics group. In particular, there's actually a strong emphasis on the intersection of neuroscience and epigenetics too, which is a bit of, more of a very specific niche field. And yet we have a lot of that here as well. So it's a wonderful community and very collaborative. So it's been a really fun place to get to start a lab in this area.
So to talk about your science, I want to start in 2015. Back then, you were first author, and you were in David Ellis' lab on a paper about BRD4, where you showed that BRD4 mediates the transcriptional regulation underlying learning and memory. So how did you get into this, and what did you find there?
Yeah, great question. So I had sort of two main papers in my postdoc, and I'll kind of lump them together to answer your question because they kind of really happened in parallel, even though one came out before the other. But when I joined the lab, I was really lucky. Bob Darnell, another lab at Rockefeller University, approached Dave right about that time and said, "Hey, we have this really interesting result where our FMRP protein of interest, which is a RNA binding protein, appears to bind to a bunch of transcripts that encode chromatin regulators. We don't really know what those are, what they do, but that's what you study. So do you want to look at this interaction and maybe in the context of Fragile X, where this protein is lost, and maybe try to figure out what's going on with us?" And that just fit perfectly with my background in neuroscience and my hope to really jump into the chromatin field in depth in Dave's lab. So I was really fortunate to kind of come at just the right time to be part of that bigger collaboration. And we got to sort of merge expertise from these groups. And the first thing we did was first just dig into some of this particular transcripts that we found in this FMRP binding protein binding transcript data set, one of which was BRD4. At the time, BRD4 was a really exciting protein. People had just figured out how to drug it, and it was one of the first reader proteins that was successfully drugged, meaning you could actually inhibit its function. And that gave me a really important tool that let me kind of go fast and quickly into this field and figure out what this protein was doing in neurons. So sort of our very first experiments of just throwing this drug on neurons yielded some really robust effects, and that launched the project. And in the end, we were able to demonstrate that blocking BRD4 function in neurons blocked activation of a number of really critical genes, particularly in response to various stimuli. And that, in turn, led to a block of memory formation in mice. So we kind of figured out this new mechanism through which a reader protein could affect information storage inside a neuron. After that, we applied that knowledge to a Fragile X mouse model that the Darnell lab worked with commonly, and demonstrated that there's actually really widespread histone and transcriptional disruptions in these mouse models of this disorder. So it turns out that if you get rid of Fragile X, for the Fragile X protein, FMRP, you wind up really aberrantly causing this upregulation of histone modifications that lead to too much transcription. And the cool thing here is this is a way that cells actually can use regulation of translation to control transcription. So it almost goes backwards from how we usually think about these things, but it's because FMRP happens to be binding the transcripts of these genes that encode the chromatin regulators that then change histones. So we found this interesting pathway that was really disruptive in this disorder. And then to try to figure out how much it really mattered, we went in and tried to block that effect. And to do that, we again used this BRD4 inhibitor that was developed around then to see if we could actually sort of treat these cells at the level of chromatin to change the cellular function. And it turns out we could. So I think we were able to kind of find a hub of dysregulation in these cells that actually controlled a lot of downstream effects. And when we blocked that particular batch of overexpressed genes, we could really attenuate many of the effects in the disorder.
So did you carry this further, so the BRD4 project, or did you leave this in David's lab?
So when I started my lab at Penn, I had a lot of different interests. And one thing I wanted to do was look beyond just Fragile X. So Fragile X is one disorder, and it can lead to intellectual disability, an autism diagnosis, and so on. But we now know there's a lot of different chromatin regulating mechanisms that, when they're disrupted, lead to similar syndromes. And I wanted to kind of go beyond just this one disorder and try to look at many others. And so that's really where we went when I started the lab. Instead of just focusing on Fragile X and FMRP and BRD4, we began to kind of expand our work to think about a lot of other chromatin regulators that hadn't been as well studied, where we thought we could really make a big difference in understanding their function in the brain and then how their disruption can lead to these syndromes. So that was more of the direction we went in, kind of building on those concepts, but going after different proteins.
You specifically looked at 5 chromatin modifiers that are all linked to autism spectrum disorders, despite their different roles in regulating chromatin, right? So this was like the first work you did in your own lab. What did you find there?
Right. So one of the first things we did was just use an RNA sequencing data set that we had and tried to just do everything we could to understand what came out of it. Somewhat because of the times, which were COVID years, where we were somewhat limited in the experiments we could do, but had a sort of really powerful data set, which, as you described, was the effects of looking at targeting not just 1 or 2, but actually 5 different genes all linked to autism spectrum disorder, and looking at their resulting gene expression changes. And what we found was that there was actually quite a lot of overlap. And these were chromatin regulators that did really different things. So we weren't really necessarily expecting that. And of course, they all had their own unique functions as well. But nonetheless, what we kind of stumbled upon was that in neurons at this particular developmental stage we looked at, there were a set of genes that seemed to just be really sensitive to any kind of chromatin disruption. So that was the sort of the main conclusion of that work, was that if you look at, say, a set of neuronal transcriptome, you can find sets of genes that are very likely to be, for example, downregulated if you disrupt any number of chromatin regulating mechanisms.
Yeah, and that would have been my next question, right? What do you mean by chromatin disruption? Because that can be a lot of things.
Of course. So in that early study, we took a kind of very brute force approach, or a kind of simplistic one, of saying, well, a lot of these variants we see in patients are often loss of function, not always, for sure, and they can be much more complicated than even just a general loss of function. Perhaps just the catalytic domain is disrupted, or just a specific binding partner is disrupted. But in this case, to be able to kind of look at things on the scale of multiple genes at once, we just took the approach of depleting the gene using a lentiviral infection with hRNAs. So a fairly simple, just knockdown approach. Although we didn't completely get rid of these proteins, we were just shooting for about 50% loss of the transcript levels to mimic something like a haploinsufficiency model in a patient. So again, these weren't exact patient variants, and there's plenty of ways you can disrupt these proteins. We took an approach of just partially depleting the overall level of the protein to somewhat modulate whatever that particular chromatin regulator was doing. And we've since become a lot more sort of precise on our methods, as well as going after very specific disorders and really characterizing individual patient variants, for example.
And maybe we can circle back to this a little bit later. But you mentioned that this was the time of the COVID years, right? And the next topic I want to talk about is the virus itself. And we already did one episode where we talked about epigenetics of COVID. However, you also looked at this and at a process that is called histone mimicry. Could you briefly explain what histone mimicry is, and then talk about what you found?
Certainly. So this was a project that really kind of happened by accident, as did many things during early COVID years. So histone mimicry is something I always found really interesting, and not at all an area that I had much expertise in at all, although other folks at Penn and other places have really done some amazing work in this field. Various proteins, both our own proteins actually, as well as other proteins, say, coming from viruses, can modify the transcriptional output of a cell by actually mimicking proteins, including histones. So, for example, there's some other viruses that can mimic very specific stretches of histones, and in doing so, often act, say, as a sponge of those proteins that are supposed to be going and targeting the endogenous chromatin machinery, and instead kind of soak them up, or pull them away, or in some way disrupt those regulatory mechanisms, changing the transcriptomic output of that cell. And one reason a virus, for example, might want to do that is because when a cell's infected, it can activate a whole downstream cascade of signaling molecules that are intended to fight off that virus. And that includes transcriptional output that will turn on a bunch of genes and then proteins that can then mount some defense in that cell. So viruses have to be really well equipped at sort of blocking these different signaling mechanisms if they want to have any chance of surviving.
So what kind of proteins are those that mimic the histones in humans from the virus?
So I can tell you a bit about the one that we happened upon. Where I actually have a little more expertise than the many others that are out there. But the way we wound up finding this particular protein was something, again, of an accident where we were all stuck at home in the first few weeks of the pandemic. And I was trying to come up with things we could do as a lab that were moderately productive or would at least keep us engaged and thinking about science. We were a really new lab. We had virtually no data to analyze. We had just sort of barely gotten the lab set up and working. And we weren't sitting on a bunch of papers we could write, for example, and really not even much data we could analyze aside from that one RNA-seq data set that led to our first paper that you already asked about. So I asked one of my lab members, a phenomenal technician named John Key, to take all the histone proteins that we study and love, and just overlap the protein sequences with the recently published proteins that had come off the sequencing of SARS-CoV-2. So, because the sequence was generated so quickly and made available worldwide, we had it at our fingertips. And I genuinely expected nothing to come of this. I just thought it would be a great way to kind of learn a new bioinformatic tool that we could maybe use downstream at some point, because one of the few things we could do was sort of learn some more bioinformatics while we were sitting on our computer, and this seemed like an interesting way to do it. And I figured, all right, well, I'll assign this task to somebody. Nothing will come out of it, but, John will learn how to do this interesting thing, and he'll give the lab a little presentation about this tool, and maybe that'll be worthwhile. And John comes back to me a day or so later, and I actually thought he was completely just messing with me because it was right around April Fool's Day, I think. And he told me that he'd found a motif that had an ARKSAP site inside of one of the very poorly understood SARS-CoV-2 proteins. And I really thought he was joking, because this was actually one of Dave Alice's favorite motifs in all of histones. And it turned out John was not joking. He actually had found this, and it was in there. And all of this could have been a coincidence, and perhaps meaningless, and just one of those things that happens upon occasion. But I said, if you're interested, you can go back into the lab and test this. We are allowed to do research if it's related to COVID. So he was game, and he and I and another lab mate started going in periodically in turns to kind of start trying to figure out if something might be happening here. And we started to see some really interesting results where just putting this one protein in to cells could drastically disrupt the histone landscape. And then that one thing led to another, and eventually we were really fortunate to be able to collaborate with experts in the field, such as Susan Weiss's lab, as well as other epigenetics labs like Shelley Berger and Ben Garcia and so on, to really expand this project quickly and use things like virally infected cells with different mutations so that we could try to understand what this protein was doing. It was one of those cases where being somewhere that has a really rich collaborative environment was so crucial, because I am not a virologist. I know very little about this. I was a new lab. I didn't have everything running yet, but we had great neighbors. We had great collaborators, and they were really willing to help throughout this entire project, which let it become a big story.
Yeah, this is what happens when you give a scientist like a task and then they get creative, right?
Exactly.
So the next thing, and this was just last year, so in 2024, you investigated the function of H2BE. So the first question was this, did you know about this histone variant, or did you discover it? And what did you find out about the function of H2BE?
So this story is one of sort of those really important ones to my group, because it was a project that one of my very first grad students started working on. And I actually was first sort of made aware of this particular histone variant, H2BE, by Dave Ellis. When I was really early days in my postdoc and deciding on projects, we were thinking about these interesting BRD4 findings and other possible ideas. The world was wide open in terms of what I could have worked on. And Dave handed me this paper from Catherine Dulack's lab that had found H2BE. And they specifically had found that the H2BE transcript was super high in the olfactory system. And they did some kind of cool functional analysis of its role there. But at the time, there was no antibody available. The tools were a lot more limited. And they had concluded reasonably based on what they had that it was probably specific to the olfactory system and not expressed anywhere else. And I didn't really want to work on the olfactory system. It wasn't my area of expertise. And meanwhile, these other projects took off quickly and effectively. So I really dug into the BRD4 and Fragile X side of the lab and focused my postdoc on that. But on the side, I kind of kept playing around with H2BE a little bit here and there and started to find some interesting things, including some suggestions that it was probably also expressed other places. And that got me more and more interested. So when I started my lab, I had a bit of data. I actually had an antibody that was specific to this protein, and that let us take off and try to figure out what it was doing in chromatin, in the brain, and so on. And then thanks to some really beautiful work from my first student, Emily, we were able to really characterize its function. Thanks to the Dulack lab for sharing knockout mice with us. So we were able to do really in-depth, behavioral and transcriptomic studies. We found out that it is able to control accessibility in chromatin. So it can really kind of change chromatin structure when it gets incorporated. And in doing so, it can promote transcription of key genes. In neurons, it turns out it really regulates synaptic genes. And that means in a knockout context, when you get rid of it, you have very weakened synapses. And the animals turn out to be very bad at forming memories as a result. They're otherwise quite healthy, I'll say, but they're very bad at storing information and kind of creating new long-term memories.
So usually when you talk about chromatin structure and chromatin accessibility, H3 and so histone H3 and histone H4 come to mind, and all the modifications at their tails. So why is this variant now important in chromatin structure? Is it because of the specific tissue that is expressed in, or did you find out something about that?
Yes. So for H2BE, it has a very specific amino acid change at site 39, which actually lies at the chromatin, the histone DNA interface. And we actually went and tested all the different amino acids in H2BE and found out that it really was this one single amino acid that seemed to contribute mostly, at least, to that chromatin accessibility change. And I'll note we saw this in both cells, so we could use things like attack sequencing to look at those changes in an actual cellular context. But we also did some biochemical assays in collaboration with others, where we could look at chromatin in really just a pure context in the absence of any binding partners or modifications. And there we could see very similar effects. So that told us it was really an innate feature of this protein itself that didn't rely on any cellular components or even histone modifications to have an effect on chromatin structure. And in turn, you can infer from that it actually probably doesn't really matter what tissue or cell type this protein is expressed in. It'll probably still have this effect because it doesn't really matter what else is there. It can do this by itself. Certainly, it's probably more complicated than just that. And I imagine a lot of other chromatin factors come into play when we think about it in a cellular context too. But we do think that this particular function of H2BE can be pinpointed to a single amino acid and is really independent of cellular factors.
So this would then just weaken the interaction of the histone to DNA and then destabilize the nucleosome?
That's certainly one model through which this could be acting. There's other ones that we're thinking about too, and we don't know for sure exactly why that single amino acid matters. Mm-hmm. There's a lot we still don't understand about this protein that we're actively pursuing in the lab now. But I will note one exciting thing is that mouse and human H2BE proteins are a bit divergent. They're not quite identical, unlike a lot of the other histones when you go across species. And I think that's one reason the field really hasn't studied H2BE proteins in a lot of depth. It's a really complicated gene landscape. And that makes it quite difficult to understand. But between mouse and human, there is a single amino acid difference that's conserved, which is that 39 site. So we do think that there's certainly reason to suspect that this particular function is conserved across species as well.
Okay. And maybe also in evolution.
Yes.
Yeah. So there currently is also a bioRxiv paper available that was posted in November 2024, so hopefully close to being published. So in this follow-up work, you investigated how H2BE is regulated by extracellular stimuli and whether it controls activity-dependent transcription and cellular plasticity. Can you talk about this work and what you found?
Certainly. So with neurons, of course, one question we almost always ask is, well, what happens when you activate the neuron, right? We certainly also care about neurons under their basal state, but one thing that they have to do that's super important is respond to incoming stimuli, which lets us do all those amazing things like think and remember and learn. And to do that, neurons have to activate transcription of a number of downstream genes and then also deactivate those genes and so on. So we actually quite early on looked at what happened when we stimulated neurons and measured H2BE levels. And what we got was actually really quite surprising. We had almost a backwards result when you think about how neurons normally respond. Neurons turn on a lot of genes in response to stimuli, including other histone variants like H3.3. H2BE does the opposite. It goes down. And this was kind of confusing, especially because we know H2BE seems to open chromatin, and that sounds kind of backwards. But the key thing was that response happened very slowly on a matter of hours, not minutes, when most sort of those fast activation responses happen in neurons. And what that really suggests is a homeostatic plasticity mechanism. And this is a type of plasticity where neurons have to kind of modulate their overall firing rate with time. So if they're getting too much stimuli, they actually need to sort of dampen their responses so they can go back to their basal preferred level of firing. They're not getting enough inputs. They have to kind of ramp it up and ramp those responses up so they can kind of maintain an active circuit in the brain. And if you don't have those mechanisms in place, you can have things like seizures. So these are really important and kind of the flip side of those fast responses that happen quickly inside of a cell. So what we looked at was whether or not H2BE may contribute to those kinds of long-term changes and these sort of homeostatic plasticity mechanisms. And in fact, it does seem to do that. So when we provide neurons with sort of a long-term stimulus, you get less H2BE. We can see that by cut and tag, by overall levels, and so on. We can see it in the mouse too. If you just put mice in an enriched environment where they can just play for a whole day and kind of run around and have fun, we actually see that dampen H2BE expression also. And then from there, we actually looked at effects on, say, electrical activity using multi-electroarray recordings and did some single nucleus RNA sequencing from brains following a stimulus as well, and see similar effects in those contexts too. And I'll just note, because these papers aren't necessarily up on bioRxiv yet or still working their way through the revision process, but this is sort of one of several follow-ups that we're pursuing now. It turns out that H2BE is also really highly expressed in astrocytes, in fact, probably more so than neurons. Another one of my first students, Sean Lozon, has been pursuing this. And it turns out H2BE also really matters to astrocytes. And this was quite exciting because very few studies have really investigated histone variants in astrocyte transcription and function. But they're quite important cells as well. And we were really excited to see that H2BE matters outside of just neurons.
Which is also very interesting and opens up probably more work in the future.
Yes. Kind of keep leading to more and more questions, but that's the fun part of science.
So we talked about this already before recording the interview. So you're working on grant proposals currently. So can you talk about what you want to do in maybe the next 5 years?
Absolutely. So we, of course, want to do many, many things in the lab. And are always thinking of new ideas and directions that are exciting to pursue. And I'll maybe just touch on a few of them. We've recently, along with a lot of clinical collaborators, found a number of patients with a neurodevelopmental disorder that have variants in the gene .1L. This is a histone methyltransferase protein that adds a modification, H3K79 methylation, onto chromatin. And there's been quite a lot of work on this particular modification by many in the field. And we do know quite a bit about it. But discovering this sort of cohort of patients, many of which have variants specifically in the catalytic domain, made us quite interested in trying to sort of tease apart the many different functions of this complicated .1L protein that adds on this modification. So we've generated a number of ES cell lines and are working with collaborators who've also made a number of useful lines to try to kind of tease apart the very different effects of this particular protein on this histone modification, but also on chromatin more broadly. And we're pursuing those mechanisms now as one main project. We're also, as I mentioned, really expanding our work on histone variants, particularly for H2BE. We're looking at the proteins that actually regulate its incorporation into chromatin. But actually, in this case, we've also started to find human patients with variants in the H2BE encoding gene that seem to affect its function in really interesting ways and lead to things like intellectual disability and a lot of growth deficits in people. So we think this actually may be another disorder that is as yet uncharacterized in people. And then in addition, we're really trying to expand our toolkit in the lab. We're applying proteomics in new ways with new mouse models to try to take a more sort of unbiased view of chromatin in the brain and figure out what and how it's responding to different kinds of behaviors and stimuli and so on. So we have quite a number of new directions as well as expanding on areas that we've already studied.
Are you also looking at interaction partners that would interact with H2BE, so that then change the chromatin environment?
Absolutely. So we are pursuing some hits we have off of a big mass spec experiment, for example, looking at its interactions and how the specific interactions might be different than canonical H2B. So what we see there are a number of shared proteins, but also some ones that much more preferentially associate with just the variant or with canonical H2B. And we're pursuing many of those as well. We don't yet understand all of them, and certainly a number of those kinds of differential interactions are going to be in some ways just a consequence of its function. So H2B tends to go to kind of open chromatin sites and maintain that open structure. It tends to go to probably the nucleosome just upstream of the TSS, so a very specific location that will have sort of specific binding partners. So we always have to think about this carefully because many specific interactions will be a consequence of its function, not necessarily part of its ability to mediate chromatin changes. But nonetheless, we're also seeing some really interesting other interactions and even specific modifications that we think are really important to its function as well that we're pursuing now.
So for the last 33 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I think I would just say that it's always a challenge to pursue unanswered questions, and it's not easy to do these hard experiments, in particular in a complex system like the brain. But it's also really fun, and I think this career is really a privilege, and I'm very grateful to have it. It's also always worth highlighting again that, I did very little of the work I just told you about. Most of it was due to the incredible hard work of a number of students and postdocs and technicians, only a few of whom I've managed to actually mention. But it really is a team effort, and more so these days. I already mentioned a few cases where our projects were the result of major collaborative efforts, but I think every one of our projects has fallen into that category so far. And I think that kind of trend will continue. So I think it's always worth remembering that what we do is really exciting, and it can be hard to remember that in the context of changing funding climates and the many challenges that we're all facing, perhaps especially these days. But that in the end, it's a noble goal to pursue these unanswered questions, incredibly important for our understanding of who we are and how our brains work and how we think and how we can continue to understand epigenetics and neuroscience and all the fun things in between.
So my last question is, what is your definition, your personal definition of epigenetics?
So I often say that I don't actually do epigenetics if you want to be strict about it, because I, not fully, but mostly work in a post-mitotic cell that does not generate new cells. So if we're going by the strict definition of epigenetics, where you're talking about heritable traits that gets passed to a daughter cell, my cells don't do that. So our neurons are in the lab. They don't have daughter cells. They can't pass on those memories, those epigenetic memories in that way. But they are incredibly important cells that have to maintain information in their chromatin for not just a cell cycle or a few days or a few weeks, but for years. You have neurons that were in your brain when you were born that will still be there when you die. And that kind of longevity is exceptionally rare. And one of the few other things that come to mind when I think of something that stable is a histone protein. Those are incredibly stable proteins. Again, the histones in your brain many of them were there when that cell was born and will be there until it dies. So I think these things go hand in hand. So I certainly favor a version of epigenetics that allows for us to think about it in the context of really lasting, stable pieces of information that are embedded within our chromatin landscape, while also being dynamic and can be modulated by extracellular stimuli. As opposed to perhaps a more strict criteria, which means that I maybe shouldn't be part of the institute that hired me.
What is it then what you are doing if it's not epigenetics? Is it transcriptional regulation, or what would you call it? Or would you call it epigenetics because of what you just said?
I do call it epigenetics at the risk of perhaps aggravating those who prefer a stricter definition. But I'm also very happy to say that we do histone biology and study chromatin in the context of neurons. Those are great words too, and certainly applicable to what we do.
So thank you, Erica, for your time and for being on this show.
Thank you for having me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.